And welcome back, everyone. Our last company for today is Rakovina Therapeutics Inc. It trades on the TSX under the symbol RKV and is focused on the development of new cancer treatments based on novel DNA damage response technologies. Please welcome Executive Chairman Jeffrey Bacha and CFO David Hyman. Welcome, everyone.
Hi, everybody, and thank you to the organizers here for inviting Rakovina Therapeutics to present at this meeting. This is really a first-time event for our company as we listed on the Toronto Venture Exchange just this past spring. As was mentioned in the introduction, we are a cancer research and development company located in Vancouver, British Columbia, focused on the development of new cancer treatments based on novel DNA damage response technologies. I'm joined on the call today by Mr. David Hyman, our Chief Financial Officer, and Dr. Mads de Gaard, our President and Chief Scientific Officer. And following the brief presentation, we'll all be available to answer your questions in a discussion period. So please take a minute. These are our disclosures regarding forward-looking statements that might be included in today's discussion. You can review additional detail and the risk factors associated with owning our securities, which are available in our public filings on SEDAR. So Rakovina is dedicated to developing innovative treatments with the potential to improve patient outcomes across many different types of cancer. As you're probably aware, as a research community, in general, we've made tremendous strides against cancer over the past several decades. But cancer in its many forms remains a leading cause of death worldwide. In 2020, there were more than 10 million deaths associated with cancer, and the market for treatment of cancers was more than $150 billion worldwide. Part of the reason for this continued growth is the emergence of resistance to currently available treatments. You can think of cancer as a smart disease whose cells evolve very directedly to outwit the treatments that we try to throw against them. And our job as researchers is to stay a step or two ahead of cancer's evolution. In Canadian terms, that's like saying we want to skate to where the puck is going to be. And that's ultimately how we're going to beat the disease. A major emerging field in this sort of skate to where the puck is going to be concept is the field of DNA damage response technologies. DDR research has become a major area of focus across the academic and pharmaceutical oncology communities. And what we've done at Rakovina is to assemble a pipeline of DNA damage response technology assets that were already under development and have been incubated independently before we came along. We've acquired the rights to these technologies that have already established proof of concept in order to leverage development efforts and optimize and accelerate our development of these technologies into human clinical trials, ultimately with the hope of seeking regulatory approval and benefiting patients. So what is DNA damage response? And at a 50,000 foot level, you think about DNA damage response as being there to protect us. Our cells literally experience insults from radiation, from toxins, from other insults, thousands of times a day, leading to defects in replication. This is happening constantly, and the DNA damage response systems are designed to seamlessly detect and repair this damage that tends to occur quite naturally. What we've come to understand is that certain types of cancer will take advantage in a defect in one or more of these repair systems. And when a mistake can sneak through and become ingrained in the cell, and if that mistake is associated with cellular growth, a tumor will form and a patient will eventually be diagnosed with cancer. What we also understand is because these errors in a DNA damage response system are unique to the cells within that tumor, we can target them with a precision medicine. And that allows us to attack the cancer and sparing toxicity generally with normal cells. Think about it like this. If we have two systems, A and B, and the cancerous cell has a defect in pathway A, leading to the formation of a tumor, but it also leads to a dependency on pathway B. So if we develop a drug that targets pathway B, it'll kill that tumor cell. But the normal cell, because it still has an intact pathway A, can survive beyond the treatment and avoids side effects that are generally associated. So this is something that's called synthetic lethality. And this allows us to specifically target tumor cells harboring defects in a DNA damage response platform. So this has been and has become and continues to be a huge area of research for big pharma. There are currently 4 DNA damage response drugs called PARP inhibitors that are approved by the FDA. Each of these are expected to become blockbuster, multi-billion-dollar level products over the next several years. And the emergence of these important drugs in the field, which are now mainstay treatments of prostate cancer, ovarian cancer, and certain types of breast cancer, led to some big acquisitions early on in the DDR research field. So $14 billion for Pfizer to acquire Medivation to get access to their PARP inhibitor in 2016. Tesaro was acquired by GSK for $5 billion a couple of years ago to get access to their PARP inhibitor. And now what we're seeing is that big pharma's investments have moved sort of upstream into early stage or earlier stage research, where they're investing billions of dollars to get access to discoveries at companies as they move forward. And we think that this really positions Rakovina Therapeutics very well as we advance our pipeline. So we're initially focusing on areas where PARP inhibitors have already shown clinical and commercial success, but where treatment resistance has emerged. So multi-billion-dollar markets like triple-negative breast cancer, prostate cancer, ovarian cancer, where PARP resistance has become a major unmet need, is where we're beginning to focus our efforts. And we believe this strategy will not only allow for accelerated development, but will also be attractive to potential partners. Our pipeline currently consists of 3 distinct molecular series, each of which has established proof of concept before we picked up the rights. The KT-2000 series is a novel series of PARP inhibitors, which is being screened against resistance mechanisms for improved biodistribution, in particular to treat tumors that have metastasized to the brain and the central nervous system. The KT-3000 series is a dual function DNA damage response inhibitor that we think of as a smart molecule. We know that when you inhibit the PARP enzyme, certain other pathways emerge to cause resistance. So this class of compounds is designed to anticipate that and be able to head that off as it begins to happen. And the KT-4000 series actually carries a payload, a targeted payload to cause DNA damage to a tumor cell's DNA and then be able to actually inhibit its repair. And each of these, as I mentioned, were previously under development by third parties before we picked them up, which we brought under the Rakovina research umbrella to really accelerate lead optimization and advancement into human clinical trials. We're undertaking our lead optimization activities in collaboration with the University of British Columbia under the direction of Mads Gaard, who's on the call with us today. We're very grateful for this relationship, which has given us access to world-class facilities and world-class researchers without the time and capital investment required to build it ourselves. This platform also gives us real-time access to data versus using a contract research organization, which we believe will really accelerate and streamline our efforts and shorten lead times as we go through the optimization process. Our portfolio, as I mentioned, is currently being optimized to select candidates for advancement into human clinical trials. On this slide, you'll notice something that may be a little bit different than what you might be used to seeing in traditional drug development, which is typically a phase 1, phase 2, phase 3 approach, where you start in healthy volunteers. In the cancer field, we're able to actually start in patients out of the gate. So from the first time we're testing the drugs in human clinical trials, we're actually looking at responses in patients rather than looking at just safety in healthy volunteers. This is pretty common in the cancer field. The other thing that we are able to do is move into a pivotal study, which can be a single-arm trial designed to show that the drug can shrink the tumor versus a large randomized phase 3 trial. The FDA has allowed accelerated approvals based on the demonstration of tumor shrinkage, which is considered a surrogate response for overall survival. It's a pretty obvious idea. If you shrink the tumor or make it go away, the patient will live longer. So this would allow us to potentially access the market prior to large phase 3 clinical trials. A great example of this is a drug called osimertinib, which started its clinical trials first time in man in 2013, and less than 2 years later was awarded an accelerated approval by the FDA in lung cancer. They still went on to complete randomized phase 3 studies with osimertinib, but that was after the drug was already on the market. And it generated over $1 billion in sales between the time of accelerated approval and full approval after that phase 3 stage. So that's the kind of strategy that we're after to really accelerate our development and unlock shareholder value rapidly. Our leadership team across the board has more than 200 years of collective experience in pharmaceutical research, drug development, regulatory affairs, and company building. It's safe to say that across this group, billions of dollars of value has been created during everyone's collective careers to date. And the detailed backgrounds of the team are certainly available on our website. Our capitalization looks something like this. Our public float is approximately 30 million shares. The majority of those are closely held or were purchased in our go public round, where we raised an initial $4.7 million to capitalize the company. We also have an additional 4.5 roughly million in available proceeds, or 5.5, excuse me, through the exercise of options and warrants. As mentioned, we raised an additional 4.7 million in our go public transaction. You can see here the majority of the expenses in the prior quarter were non-cash startup related expenses and transaction costs. David, our CFO, can go through this in more detail during the Q&A if anyone would like. The bottom line is we ended the quarter, ended March 31st with about 4.6 million in working capital, which really positions us with a lot of horsepower as we advance our near-term goals in our research and development programs. So with that, I will conclude the formal remarks and open up for questions, and thank everyone kindly for your attention, and we look forward to a fruitful discussion. Okay. I'm not sure where we see the questions come through here. Okay, I think I'm seeing a first question come through here.
Hi, everyone. Yes, I'm sorry. I'm having some internet issues where I'm staying, so I'm ready for these questions. I'm so sorry. All right, we'll start with the first question. Can you provide us some more background on the experience and past successes of your Rakovina Therapeutics and the leadership team?
It looks like I got dropped out of the list. I'm back.
Sorry. So who would like to answer that one? We're talking a little bit about the therapeutic leadership team.
Yeah, I think I'll let Mads and David introduce themselves briefly, and then I can add some color to that. Mads, I think you're on mute.
Sorry for that. I can start. So the therapeutic leadership team is comprised of me as a CSO. I'm also a professor at University of British Columbia, and I have a background for more than 15 years working with DNA repair and genotoxic stress in cancer. And we are conducting all the preclinical work at the UBC in our animal facilities and our technical course, together with Sarah, that is also on the call here, and who is a project manager of the team.
Okay, thanks, Mads. I'll jump in next. I'm David Hyman, CFO of Rakovina. I'm a chartered accountant and chartered business evaluator by trade. 20 plus years of financial and accounting experience, including stints in public practice at Deloitte, capital markets as an equity analyst at Raymond James, and time as a private equity analyst at Camcor Partners. Most recently, I've been the CFO or primary financial executive for 4 startup companies spanning fintech, e-gaming, cannabis, life sciences. Through that time has included multiple M&A transactions and 4 go-public transactions.
And this is Jeff. I'm the executive chairman of the company. My background is originally biophysics. Spent a little bit of time years ago on the benchtop before pursuing an MBA. Was recruited to my first company here in Vancouver, British Columbia, a little over 20 years ago. Took that company from a valuation of a couple tens of millions when I joined to about 700 million over the course of about 18 months. As part of the team there, involved in multiple startup companies, most of which are now publicly traded on the NASDAQ, and are looking forward to building up the Rakovina pipeline and team to do the same thing. Just as I mentioned, the people around us collectively, we've got hundreds of years of experience. People like John Langlands, who's got 25 years plus of operational experience in the pharmaceutical industry, and he actually led academically the establishment of the drug development program at the University of New South Wales in Australia. Dennis Brown started Matrix Pharmaceuticals, started Chemgenics, both of which were acquired following their NASDAQ listings. And Dennis and I also founded a company called Delmar Pharmaceuticals, which is now Kintara, and has a drug in pivotal clinical trials for brain tumors. Neil Sankar, our acting chief medical officer, has led programs at big pharma and smaller companies, oversaw the development of Clydecla and ibrutinib through FDA approval in the cancer field. Len Post, an advisor, was the architect of the program at BioMarin and Medivation that was acquired, the PARP inhibitor program that was acquired for $14 billion by Pfizer a couple of years ago. Julie Charrington was a leadership CEO of Arch Oncology and Pathway Therapeutics. And Wang Shen goes without saying is quite a famous chemist who's got a number of FDA-approved drugs on the market. So we've got a great team around us, and we're very excited to get Rakovina going in the last few months and do some great things.
Wonderful.
Next question.
Do you all see a future for PARP inhibitor in cancers that do not harbor a deficiency in HR repair? And how might your company's approach address these instances?
I think I'll point this to Madge to give an answer on that.
Yeah, thank you for that. That's a good question. So far, the PARP inhibitors has mainly been used in very specific circumstances where tumors or tumor cells, they have a defect in a specific DNA repair pathway called homologous recombination. And we believe that with our KT3000 and KT4000 series, we can move beyond those indications by using this smart molecule technology that we have in our pipeline, where we both target PARP as an enzyme, but we also target other key enzymes that are important for resistance and also important for the DNA damage response.
So there are approximately 30 million unescrowed shares in the RKV public float. Can you describe the makeup of these shareholders and how many are held by insiders?
David, why don't you take that one?
Yeah, sure. Thanks, Jeff. So the non-escrowed shares, I think we conceptually put into to 2 buckets. The first bucket is about 23 million shares that were issued in conjunction with our public listing transaction, and those were all issued at 20 cents per share. So, and we believe that this group of shareholders is generally supportive of the long-term prospects of the company. Our analysis to date indicates that there's been very little turnover amongst this shareholder group. And then the second bucket is approximately 5 million shares, which track back to the initial public offering of the legacy public vehicle, which was called Vincero Capital. These shares were issued at 10 cents, so slightly less than our offering, and are broadly held in small blocks of shares, which is usually the case in these shell entities. Our analysis indicates, again, that this shareholder group has experienced the most turnover since we started trading on April 1st. And so most of these shareholders have turned over into kind of fresh hands, if you will. So overall, we believe that the stock has been supported through buying by supportive shareholders close to the company generally. Based on trading patterns and volume, which we've been looking at, we think we've chewed through the most of the cheap stock, providing a great entry point for new investors going forward here.
Fantastic. Thank you, guys. So what's your expected quarterly cash burn rate over the next year or so?
I think, David, that one's for you too.
Yeah, good question. Something we pay close attention to. As Jeff already mentioned, when we closed the initial financing, we had about $4.7 million of working capital, which put us in a great position in about 24 months of cash flow burn based on our projections. The one thing I would point out is the most material cost to the company is our agreement with UBC, which is payable in 6 months installments through to September 2023. The reason this is important is the fixed nature of this contract really allows us to manage our costs, our research costs very precisely, and gives us good visibility on what our monthly burn rate is going to be. So over the next 12 months, we do anticipate our cash burn rate to be about $100,000 per month, but this does exclude any potential proceeds from grant funding that we're actively searching out as well.
So why did you all choose to recently list the company on the Toronto Venture Exchange?
So I think this is really a strategic decision that was based on speed and access to capital. As we were putting together this standalone company based on DNA damage response technologies and looking at the quality of the portfolio, it's really, we believe that traditional venture capital would have been a pathway that we could have gone down successfully to capitalize the company. But as you know, that often takes quite a long time to go through that process. And because we believe the portfolio positions us with a leading edge in certain aspects of the DNA damage response field, moving quickly was important. And so the going through a merger with a shell company that had some cash, doing a private placement to really capitalize the company strongly could let us move very quickly to get that done and stay basically ahead of the curve where we want to be.
So your company's affiliation with the University of British Columbia is pretty impressive. How did Rakovina Therapeutics establish this as such a young company, and what do you perceive the advantages of the relationships to be?
Yeah, so we have a very strong relationship with UBC and other leading academic research institutions based on years of prior interaction and relationships and projects working together across a number of companies. So, putting this together with UBC really let us hit the ground running, as I said, with a dedicated world-class laboratory infrastructure that would have taken significant time and investment to build. And over the years, our practice has really been to align ourselves with top academic institutions and collaborators, and we're just continuing to build on that. It was great that we were able to work with UBC to allow Mads or Professor Degard to take on a leadership role in the company as well and lead the team there. It's really truly a wonderful and symbiotic relationship that we're looking to grow.
Great. Talk a little bit about your 12 to 18th month milestones and how you'll report your progress.
Well, our goal is really to, across these three unique DNA damage response inhibitor series or platforms, identify one or more lead compounds that's suitable for advancement into human clinical trials. And we expect that process to be ongoing over the next 12 to 18 months, which would put us in a position to initiate human clinical trials likely within the next 24 months, possibly sooner, but that's the sort of the outside goal. It'll be our general practice to report our results of our research at peer-reviewed scientific meetings, and we find this is a great way to gain the attention not only the medical community but also the pharmaceutical partners that we want to work with going forward.
Will you present at cancer research meetings?
Yes, as I just noted, this is really an important component of our communication strategy. Not only does it give us access to real-time access to pharmaceutical partners and medical researchers who have similar interests, but presenting data in a peer-reviewed setting really also provides strong validation of its importance to shareholders in the broader investment community. So across the board, that's been our strategy, and scientific research meetings and publications in peer-reviewed journals are very important to us.
So do you expect to be able to leverage your working capital through grants or other forms of non-dilutive fundraising?
Absolutely. Canada has a number of very important programs that we will seek to leverage, as does the international community in the United States and Europe. And so we'll look at leverage in any ways that we can get it in a non-dilutive sense.
Talk a little bit about Rakovina Therapeutics' priorities within the development pipeline. Does one class of compounds stand out at this time?
It's interesting. We had a research team meeting recently where we kind of went around the room and said, who's got their favorite of the classes? It's really a bit early in the process to say, is there a best or a lead series? What I can say is that we believe that each series, the 2000, 3000, and 4000 series, has an element of novelty that is potentially very exciting, and that we're seeing some very interesting data coming out of the lab. So our team's excited, and so stay tuned as we have opportunities to present that data to the world.
Talk a little bit about the limitations of existing PARP inhibitors. How does your approach lend itself to overcoming them?
Well, Matt's touched on this earlier. I'll start, and then maybe Professor Dugard can add some color. There are currently 4 FDA-approved PARP inhibitors, and like I said in the prepared remarks, each is expected to be a blockbuster product in its own right. That said, there are limitations to those treatments, particularly as related to the emergence of resistance and the fact that only certain cancers, as Matt's described, lend themselves to treatment of PARP inhibitors. So our pipeline of novel DNA damage response technologies is really designed to address both of these from different angles. And Mads, I'll let you add anything else.
Yeah, no, I think you said it very precisely. If we want to move beyond what the space where the PARP inhibitors are used currently, we need to think in new terms, and that's what we are doing. We are trying to develop new compounds that can bring the PARP inhibitors outside the current use field.
So have any of presentations from recent major cancer meetings like AACR or ASCO impacted your strategies?
So I think that one thing to note is that there's a lot of real estate at these major meetings being dedicated to DNA damage response technologies, both in terms of the number of presentations, the fact that these DNA damage response research is being highlighted in plenary sessions. This really shows the importance of the field, and that it's a field that we're very pleased to be at the center of as it evolves, and is very exciting. I think we noted that AstraZeneca's strategy in focusing on PARP1 specific molecules as a way to manage potentially side effects is interesting, and something that we're watching. The emergence of data from the WEE1 inhibitors in the context of DNA damage response is interesting, and something that we'll continue to watch. Ultimately, we're working at our own data coming out and having opportunity to share these data at research meetings on our own accord. So a lot of exciting stuff out there, and we're happy to be kind of right in the middle of it.
So when you acquire the rights to the technology, is there any joint venture associated with that?
So in the 2000 series, there was, we acquired that straight out. So we own that 100%. The 3000 and 4000 series, when we started looking at them, they were initially a little bit earlier stage, and rather than jumping in with both feet, we entered into a research and collaboration agreement with the inventor, which gave us the rights to conduct our own work. And if we like what we see, we have the exclusive right to negotiate the worldwide license or acquisition of those technologies. So it's a really nice way to not enter into a transaction until we have all the answers we want to have. So we have exclusivity across all of them. The patent life on all of them is quite long. There is composition of matter claims on all of them. We have an exclusive option on the 3000 and 4000 series, and we own the 2000 series.
Do you have any patents?
As I just touched on, there are global patents filed across all 3 series, claiming composition of matter, which means that's ownership of the molecules themselves, which is the gold standard. You want to own, it's like a mining company. You want to own the real estate that you're digging within. There are a number of patents that have issued globally around the 2000 series. We announced in April the issuance of a Canadian and European patent around the 2000 series. And the 3000 and 4000 series are going through the process, and we expect to have issued patents there over the next months and years.
Fantastic. Talk about your timeline, if all goes well, to get FDA approval.
So, that's always a tricky thing to put a pin in from a lead optimization standpoint. So as I said in the remarks in the presentation, we expect to go through our lead optimization process over the next 12 to 18 months. From the time where you have a lead molecule in hand and advancing it into formal toxicology studies, that's about a 6-month window before you start clinical trials. And then it really depends on the tumor and the trial designs and how long those clinical trials would take. The example of osimertinib that I raised, that drug, which is targeted for lung cancer, started its clinical trials in late 2013, and before the end of 2015 had received its accelerated approval. So we're looking to move as quickly as we can. The important thing to keep in mind is that the value that can be unlocked through the process of lead optimization, entering into clinical trials, having clinical data, potentially partner with a pharmaceutical company is tremendous. If you look at our valuation today versus a typical valuation for a cancer company in early stage clinical trials, it's really potentially an order of magnitude difference, potentially 75 to hundreds of millions of dollars in difference from where we stand today. So, we're really looking forward to going through the process, to completing good quality research around these compounds, advancing drugs to the clinic, unlocking value, and hopefully benefiting patients significantly as we go forward.
What's your current monthly burn, and how are you capitalized, and how much more money do you estimate you'll need to raise to get cash flow positive? Talk a little bit more about that. And then are you going to uplist to any other exchanges?
So I think David talked about the expected monthly burn rates, on the order of about 100,000, give or take, a month in a previous question. The nice thing is that the relationship with the UBC is essentially a fixed cost. So we know what that's going to be like, at least for the next couple of years. As we advance into clinical trials, obviously, things get a little bit more expensive, and we do anticipate raising more money. As I said in the presentation, there's about between $5.5 and $6 million available to us through warrant and option exercise. So that's already considered to be part of the fully diluted capitalization of the company. But clinical trials tend to require even more horsepower than that. So we would look at raising additional capital prior to entering into clinical trials, in the next sort of 18 to 24 month time horizon. In terms of an uplisting, obviously starting on the venture exchange is a great place to be, gives us access to capital and visibility in a very timely manner, as I mentioned. But as we move forward toward, clinical studies and attracting institutional investors, I think it's likely that we would seek a senior exchange listing, whether it's the senior exchange in Canada on the Toronto Stock Exchange or through NASDAQ in the United States.
Any closing remarks?
Well, I'd like to thank everybody for your attention and really enjoyed our time with you all today. And on behalf of our team, we look forward to sharing our success with you as we go forward. And please feel free to reach out to us through the website if you have any further questions. Thank you.
Thank you so much for your presentation. It's extremely important technology you guys have. So thank you for joining us on the conference today.
Very good. Thank you.
Thank you.
All right. Thank you all, everyone. It's been a great day here at our Emerging Growth Conference. We hope that you've enjoyed. Now, stay with us because in just a minute, you're going to be redirected to the registration page for our next conference. So stay on to reserve your spot early. On behalf of all of us at Emerging Growth and Investors Hub, I'd like to thank you all, thank all of our presenters, all of our attendees for helping to make this first Emerging Growth and Investors Hub joint conference such a great success. Remember, there's a complete replay of this conference separated by company. It'll be available on the Emerging Growth Conference YouTube channel. That's youtube.com/Emerging Growth Conference. Please subscribe and follow us on Twitter at Emerging Growth C. We post all new information on Twitter first. So I'm Anna Berry, and everyone who made this event possible today, we wish you a great rest of your day, a successful week, and we'll see you back very soon. Thank you all for watching.